So obviously I'm not here again today, so Mr. Ing is here, and you guys know the deal. Stop if things need to be stopped, if Mr. Ing needs to stop stuff, whatever. So we're going to talk about mitosis and meiosis, which is necessary for reproduction, because yesterday we dealt with sexual and asexual reproduction. So we might as well go with the how does that actually happen? So we're going to look at chromosome numbers in terms of our question. So when our cells replicate, basically what you need to do is a few things. First thing you need to do is whatever's inside of the cell needs to double. Then we need to double the genetic information. And once we've done that, we can then split everything apart. The catch is when we split everything apart, we have to make sure that we have equal amounts of things in both cells. And that includes having the equal numbers of chromosomes, meaning we number our chromosomes 1 through 46. Well, technically 1 through 45, then we have an X and a Y or an X and an X. So by the dividing up, what I mean is each cell will have a chromosome 1, each cell will have a chromosome 2, each cell needs to have chromosome 3, each cell has a chromosome 4, so on. The reason for that is if you're missing some chromosomes, this might be bad because those cells or those chromosomes are probably essential. And you also run the risk of if you have too many chromosomes of one particular kind, there's something called a dosage compensation, which is actually a pharmacology phenomenon, which is basically the poisons in the dose. Meaning, if you receive too much of something, it tends to be lethal. So water is good for you. Too much water will kill you. So it's not that water is good for you, it's how much water are you receiving. That is the part that ends up answering the question. So if we look at the simplest of the organisms, so this isn't just isolated to to prokaryotic organisms, because there are definitely eukaryotic organisms that do this too. But the easiest thing that we can do is what's referred to as binary fission, meaning double everything that you need to double, and since if it's dealing with a prokaryotic organism, there aren't too many cell parts to have to worry about. So we just basically double the genetic information and we'll split the cell in half. That's the end of the story. So this one is really nice, easy, simple. Namely because you just go, we don't have to have specialized cells or anything worrisome like that. So for prokaryotes and a handful of eukaryotes, it's not that bad. All they have to do is receive signals that say it's okay to reproduce. So signals will bind, that will cause a transduction cascade, and the result is we divide. With us being eukaryotic organisms, we have a little bit more complicated of a path. And the first bit of complication is our DNA is not DNA. When we look at our chromosomes, it's not just a bunch of DNA. It's actually a material called chromatin. Chromatin is a mixture of proteins and DNA. The name of the proteins are called histones. Well, among those proteins are called histones, which are just little bundles that the DNA wraps around, and you create these little bundles here called nucleosomes, and then these nucleosomes start to all wrap around each other, and then those bundles start to wrap around each other, and eventually what you end up making is a thing that we would call a chromosome or a metaphase chromosome. When we look at a cell that's not dividing, all of the chromatin is loose. It's just kind of spread out everywhere. And it's only during replication that we actually see it all become condensed, because that's when we actually have to start shuffling it around. So chromatin, our DNA, our chromosomes, actually move between two different states. One state of being all super compact and another state of being spread out. So just because there's some terminology with this, it's not that it's super duper important that you can remember all of it, but it will make your life easier. Is if I have DNA by itself, it's called DNA. If I have DNA plus protein, namely histones, we call it chromatin. If I look at chromatin, it can be put into a thing that we call a chromosome. Whether you see the actual chromosome or not, it doesn't matter, it's still chromatin. Here becomes the thing. So chromatin can show up in a uncondensed form or condensed form. The chromosome is either condensed or uncondensed. When I look at a chromosome after it's divided or after it's replicated, what you actually get is this X shape. So this X shape is actually because it's already undergone DNA replication. Each side is called a chromosome. So I have one chromosome, one chromosome, one chromosome. But as long as these two parts of the chromosome are connected, I also call each half a chromatid. So it's confusing. So all of this is all of this is a chromosome, all of it is chromatin. As long as these two halves are connected, each half is called a chromatid with a D. The moment I separate these two, I don't call them chromatids anymore. I would call them chromosomes. The word chromatid is when they're connected together, when you have the X shape. Each half of the X is a chromatid. I didn't come up with the words. If you're angry, not at me, please. So some more terminology are the terms haploid and diploid. We actually can go more than this, but we'll just go with haploid and diploid. So if we're dealing with us, 46 chromosomes, which are 22 pairs, what we would have is if you happen to have two of each pairing, you would be what we call a diploid cell, meaning you have two of everything. And the way that we abbreviate that is we write 2N. If you have half, meaning you have one chromosome 1, one chromosome 2, one chromosome 18, one chromosome 21, whatever, you're called haploid, meaning you have half the numbers. The way that we symbolize that is we write an N. So diploid is 2N, haploid is N. If you were a tetraploid, you would be a 4N. If you were a decaploid, you'd be a 10N. If you're a triploid, you'd be a 3N. I point that out just because that happens. Not necessarily in animals, but definitely with other organisms. Plants are very famous for not being either haploid or diploid. The reason why these exist is if I were to take two haploid cells and stick them together, I make a diploid cell. If I were to take a diploid and a haploid, I can make a triploid. These are our units of addition. So they're going to turn out to haunt us in a bit. The thing with these conditions is if we don't happen to have the right numbers, meaning if we humans don't happen to have diploid numbers for everything, we tend to get issues. So there's a condition referred to as Edwards syndrome, which is a triploid or triploidy 18, meaning you have two of everything except for chromosome 18. Chromosome 18, you have 3. The result is you're dead very soon, as in it's within like a year of life, you're dead. And you have all sorts of things that go wrong. Just chromosome 18. Patau syndrome is a triploidy for chromosome 13. So I think this one you're dead in 6 months. So you're adding one extra chromosome and the result is death. Klinefelter's is actually you have an extra X chromosome. That one you can actually compensate for. There's a few things that go wrong with you, but you can live a normal life for the most part. If you have this one, you're sterile, but you can live a normal life. There's actually another famous one called Downs syndrome, and that's when chromosome 21 turns out to be triploid. There are 3 chromosome 21s and everything else happens to be in pairs. The point is, we have the addition of just one extra chromosome, and the result is whole bunches of things going wrong with you. And that's because of the dopage compensation. We need to make sure we have the right number of chromosomes, and we need to make sure we have the right chromosomes, because things tend to go wrong the moment you are missing one or you have too many. That turns out to be the case almost all the time. The weirdo exception is the X chromosome, and that's because the X chromosome, its functionality is actually rather odd, and we don't talk about it. So in order for us to get to either making another diploid cell or another haploid cell, we have to go through a process that we call the cell cycle. The cell cycle has a whole bunch of different stages in it that are rather distinct. Some of them we can't see, some of them we can see, but we can detect them in other ways. They're called G1, S, G2, and M phase. The G phases are called growth. So this is when their cell is growing, meaning it's getting ready to do something. S phase is for DNA synthesis or DNA replication. So that's opening up at an origin of replication, adding primers, adding onto a free 5' end using DNA polymerase, or if we're on the lagging strand, you have to add a primer, which gives you a piece of RNA. Then we can start using DNA polymerase in order to extend it out, and then we have to go and replace the primer and making Okazaki fragments, all that stuff that you have sworn you would forget. Yeah, it's back. Then we have M phase, which is the actual division phase. So all of this time here, G1, or this thing that we call G0, meaning you're not dividing, you're just kind of there, not dead. So the two G phases, or the three G phases, plus the S phase, all of this is given the name of interphase, which is this I down here. Interphase is a nice way of saying we can't really visibly see what's going on. Something's going on, we just can't see it. Okay, cool, fair enough. M phase is when we can see things going on. So that's why it's kind of pulled out off to its side. Interphase, it's just there. M phase, ooh, look, stuff is going on. It doesn't mean that there aren't cellular events occurring during S phase. It's just we can't see them with a microscope. We actually have to do other things to figure out what's going on. So the cell cycle actually is divided up into two parts, or two different versions, not two parts, it's two different versions. So when you go through interphase of S1, getting ready to replicate DNA, S phase, replicate your DNA, G2, get ready to divide the nucleus, M phase comes in two varieties. One of them is referred to as mitosis, one of them is meiosis. They both have different outcomes. Mitosis, what you start with is what you end with. You're making clones. This is asexual reproduction. This is when the times are good, or we just need to have more of you, or you being that cell type. Mitosis is good for let's make more copies. Exact same copies. Meiosis is an evolutionary oops of mitosis. And what that turns out to do is it results in actually 2 rounds of mitosis. The catch is, in the first round, we don't do too much, but in the second round, we actually split the cell in half. So we actually divide up the nucleus. The result is meiosis gives us 4 cells that are all genetically different for a whole bunch of mechanisms, while mitosis gives us 2 cells that are the exact same. If I were to take mitosis and add another round of division underneath it, you effectively get these 4 haploid cells at the end of meiosis 2. You also notice that we divide it up and we call it meiosis 1 and meiosis 2. If you're curious why there's this little arrow at the top, it's because DNA replication, meaning we're doing stuff beforehand. So this 2N goes through 1 round, then another round. This 2N actually just goes through 1 round of division. Mitosis again, make us the same. Meiosis, we're going for some type of increase in variation. Forms that those gametes or haploid cells. So if I look at mitosis, there's a whole bunch of words. You're looking at this and saying, oh no, do I need to memorize all these words? The answer is no, not by any stretch of the imagination. I don't care if you know prophase, metaphase, anaphase, telophase, or cytokinesis. Although the names actually do tell you a lot of what's going on. Prophase tells you it's the first metaphase, meaning I can see a change happening. Anaphase, ana means apart or again. Telophase means far away. Cytokinesis is the movement of the cell. So the names actually do tell you what's going on. But what do? We take all of our chromatin, we'll condense it into chromosomes that we can see, prophase. In metaphase, if I had to divide up all of these cells, we have to make sure I have divide up all the chromosomes. So I have chromosome 1 in one cell, another chromosome 1 in the second cell, chromosome 2 into both cells, chromosome 3 into both cells. How would I go about doing this? Well, the easy thing would be line them all up, chop them in half, and then just start to split them apart. And that's exactly what happens here. You might not line them in the middle and chop them apart, but you would sit there, take each one, split them apart, and then move them into separate piles. The way the cell does that is all the chromosomes will be lined up in the middle in what we call the metaphase plate. And actually a thing called a spindle, which is actually a whole bunch of microtubules, will grow and attach themselves to the middle portions of the chromosomes. Those middle portions are called centromeres, meaning the part in the middle or the part that's central. You'll notice that that's actually not true. They actually don't grab the centromere. There's actually a whole bunch of proteins on the outside called the kinetochore, but you don't need to know. So these proteins, these microtubules, will grab a hold of the middles, the centromeres, and then when everything seems balanced, there's a signal that's sent, and the result is the microtubules will actually start to pull. And when they pull, they'll split apart the chromosomes, or what we'll say is we'll separate the sister chromatids, and the chromosomes will move to opposite sides of the cell. When they do that, what we can start to do is uncondense them again, add a nucleus, put cell parts back in, and then what we have to do is split the cell in half down the middle. And the result is we have 2 brand new cells. That's all that happens during mitosis. We take our chromosomes, line them in the middle, split them apart, divide the cell in half. Ta-da, done. Because we separated all the chromosomes correctly. This is what it looks like.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates.
That's not true. We know why. It's an old video, but it works.
Accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating 2 new cells in this ongoing cycle that sustains all life.
Exciting. Meiosis is basically the same thing, just done twice. So what you would do is you take your chromosomes, but instead of having them line up in the middle, they pair up. Because if they're paired up, what we can do is pull the pairs apart. Meaning, I'll take both chromosome number 1s, stick them together. Both chromosome number 2s, stick them together. And I can separate the pairs. So I have a chromosome of 2 chromatids of number 1 moving to one side of the cell, number 2 moving to one side of the cell. So that's one round. Well, we'll split apart the pairs of chromosomes. Then meiosis 2 is basically like mitosis. We'll stick the chromosomes in the middle on the metaphase plate. We'll split apart the sister chromatids in anaphase, or just we'll split them apart, and the result is we actually end up forming 4 cells. Here becomes the catch. I don't necessarily know how these cells are going to split apart, because remember, or how these chromosomes split apart. One of these chromosomes, so this red one or this orange one of those came from the mom and one of them came from the dad. The dark blue and the light blue, one of those came from mom, one of those came from dad. Here, the 2 darker sides are on, or the 2 darker chromosomes are on the same side, the lighter chromosomes are on the same side. But why does it need to line up that way? Why can't these 2 flip-flop? It's possible for them to flip-flop, meaning it could be the dark red one and the light blue one at the top, the orange one and the dark blue one on the bottom. I don't know how these are going to arrange, and the result is we get variation. There's another thing that goes on, which I think I have on the next slide. Do I have it in here? I'm going to put it here. And it's this phenomenon that we call crossing over. So what happens is when we pair up chromosomes, meaning a chromosome 1 with a chromosome 1, one from mom, one from dad, through a whole bunch of mechanisms that we don't necessarily care about, what we can actually do is reshuffle parts of the chromosomes, meaning the chromosome from mom and the chromosome from dad don't end up being only from mom and only from dad. The parts actually exchange. So each sister chromatid is going to change its parts. And it's going to do that with the other partner. So when you look, they actually form chiasmas or X's or crosses, and parts actually trade locations. And the result is they're not all the same. We've increased variation. I don't know how these are going to line up. I've increased variation. There's a few other mechanisms that show up here too, but those are two things that we can obviously see that occur that increase variation. Another video.
The cell prepares for meiosis just as it does for mitosis. But as the DNA condenses, each chromosome finds its homologous counterpart. These matching pairs of chromosomes hold equivalent genetic information, one set from mother and one from father. Next comes the first shuffle of nature's genetic deck. The two homologs trade genes in a process called crossing over. This forms new hybrid chromosomes. The spindles then place maternal and paternal chromosomes randomly on each side of midline. It is another shuffle that can lead to a host of different genetic outcomes. Finally, spindle fibers pull the homologs apart. This leaves the sister chromatids intact. With twice the amount of genetic information the cells will need, nature must cut the genetic deck in two. This second division produces 4 unique sets of chromosomes. Because they carry half the genetic information, we call these haploid cells. Many of them will mature into sperm and eggs, the reproductive cells called gametes.
Exciting. So location turns out to matter, as the video alluded to. All cells in the body are capable of division. They're all capable of mitosis. Doesn't mean they all do, but they're all capable of it. Meiosis is actually restricted inside of the human body or animal bodies, actually most organisms' bodies, to certain locations. We, animals, call them gonads, meaning if you're a male in the testes, and if you're a female in the ovaries. But even then, not all cells in the testes turn out to undergo meiosis. Not all the cells in the ovary undergo meiosis. There's actually only certain cells within these two locations that ever undergo meiosis. All the cells everywhere, including ovaries and testes, undergo mitosis. But the meiosis thing is actually very, very, very restricted. And there's a reason why. The question is, can you think of it? And as a hint, it has to do with with DNA replication, because we, eukaryotic organisms, have an issue with DNA replication. So meiosis is actually the freak exception. It's not the rule. Typically what we'll see happen in sexual reproduction is meiosis will occur to form what we call an oocyte and a spermatocyte. Oocyte's not oocyte, it's pronounced oocyte. That's an egg. Spermatocyte, sperm. What we get is meiosis happens, the two fuse, we'll call it fertilization, and the result of that is mitosis. Meiosis happens once for females, once for males. Actually, it happens a whole bunch, but we need one event where we fuse the female gamete and the male gamete. Those two fuse, then it's mitosis all the way until reproduction, cell forming time, until it's gamete forming cell time again.